Okay, so today is the fifth session of Seed Science and Technology, where we will be studying about seed dormancy. So in the all the previous classes, we started with the development of a flower, and then we have just went through the different process or mechanisms of seed maturation, drying, desiccation tolerance, and overall seed development. So after that, we have talked a few aspects of germination. So we have left out with some other topics or some topics of germination, which are different types of germination, their examples and all. So that I will be discussing after seed dormancy. So for now, we'll stick to the seed dormancy and we'll try to understand what is seed dormancy, what are the different categories under seed dormancy, and what are the different types of classification that were given, and also the which are the methods that are actually used to break the dormancy. And also in between, we'll be covering the factors that affects the seed dormancy. Okay. So we'll just start with the very basic thing. So what do you understand by the seed dormancy? Yes, anyone? I believe like you all will say about the favorable and non-favorable environment, unfavorable environment, right? So just tell me like what you know understand about seed dormancy. Yes, anyone? Just tell me what you know. So there are two terms. One is seed dormancy and second one is quiescent state. So these are the most confusing type of terms that are used in the seed dormancy. So can anyone tell me the difference between the seed dormancy and quiescent stage or quiescent state? Okay. See, whenever we talk of dormancy, it is nothing but like a failure of a seed to germinate. So when we talk about germination or when we speak about germination process, if you sow some seeds and they won't be able to germinate. So what is germination? Germination is nothing but protrusion of radical. So once the radical comes out of the seed, that process we call it as germination, right? So if this doesn't occur in a seed when you sow the seeds, so this is because of either seed dormancy or quiescent state. So what is the difference between seed dormancy and the quiescent state then? So how can you differentiate between these two? Which one, like whether the seeds are actually in the quiescent state or the seeds are in the dormant state? How can you differentiate it? The video is off. You mean like my screen is not visible to you? Yes, like okay. Just what about others? Is this true for everyone? Like you are not able to see my screen because I have shared my screen already. Not visible. Fine. Okay, sorry. So what about now? Can you just see my screen? Yeah. Okay. Fine. Okay. Thank you. So I had to ask it before, but I thought like anyhow my screen will be visible. Fine. So I was talking about seed dormancy and the quiescent state. So I told you the meaning of seed dormancy. What is seed dormancy? So when you just sow the seeds in a field or in laboratory conditions or in any media, if the seed will it doesn't have a ability to germinate, and also what is germination or what is the ability to germinate, how we see it in terms of radical protrusion, right? So if that doesn't occur, then we say that germination process is not actually occurring. So if this is the thing, then it is called as seed dormancy or it might be quiescent state. So when you sow the seeds either in germination media or in case of field, if the seeds are not germinating, it is either because of quiescent state or because of seed dormancy. So if both the things are the same, like in the both the cases, if seeds are not germinating, so how do you differentiate between these two different mechanism? One is quiescent and one is seed dormancy. Yes, can anyone tell me, like, what, how can you differentiate between these two? Okay, so let me first make it clear for you what is dormancy. So dormancy is nothing but inability of a seed to germinate when the external factors are favorable, or a failure of viable seed to germinate even in the favorable conditions. So this is what dormancy means. I hope like you got the thing. The seed dormancy means when the seeds are kept in the favorable environment, so even then they don't have the ability to germinate. So those type of seeds are categorized as dormant seed. But in the second case, when I talk of quiescent form, quiescent state is the one where it is only because of unfavorable environment. So when they are kept at an unfavorable environment, then they won't germinate. If they are again reversed back to the favorable environment, so those will be germinating. If this is occurring when seeds, if you have two different medias, so in one media the environment is not favorable or unfavorable, so in the second media if the environment is favorable, so in the first case if the seeds are not germinating, and if they are germinating in the second case, so these type of seeds are classified as quiescence, or the state of the seed is quiescent state. But if in the both the cases, if they are not germinating, then it is called as seed dormancy or dormant seed. Okay, so I can just explain this in another simple way. For example, if there are certain seeds, okay, so you want to sow them. So what actually matters for the germination? One is internal environment in terms of seed, like the environment of a seed, right? And the external environment. So external environment includes light, temperature, moisture content, and soil condition, or just the media conditions. So all these things will matter, and also the RH. So these are the few important factors that will have an impact on the germination. So these are all the external factors. So or this will come under the category of external environment. So when we talk of internal environment, it is basically the enzyme activity and the physiological state of the seed, and it's about the embryo growth and the enzyme. So okay, enzyme already written. So these are the factors of the internal environment, and also it covers the seed coat, the structure of the seed coat. So these are all the things which are under internal environment. So if this is the case, when the external environment is given in terms of, like, or given accurately, that will favor the germination. If this is done, so at that time if the seeds would germinate, they are called as dormant seed. What I mean to say is, so internal environment is fine, all the things are fine in the internal environment, but you only change the external environment. For example, in the first case, if this is the case 1, so you will provide an environment that is favorable. I will just write it as favorable environment. So in this case of favorable environment, you are placing the seeds, though they are not germinating. So those type of seeds are called as dormant seed. So the case 2 will be, you will be providing the unfavorable environment of the first place, and you will just sow the seeds. So if they don't germinate, you will collect those seeds. So you can't decide just by giving the unfavorable conditions whether the seeds are dormant or not. So you should also give the favorable environment for it. So if they are germinating in the favorable environment, so the seeds are actually in the quiescent form. This is not actual dormancy, this is just a quiescent form, because it is completely dependent upon the environment, that is the external environment, not by the internal factors of the seed. So this is what quiescent stage is, or quiescent state is. But when we talk of seed dormancy, it is mainly because of seed's internal environment. internal environment might be of embryo or seed coat or other physiological activity of seeds, which is actually inhibiting the process of germination. I hope this is clear. Just give me a confirmation, yes or no, so that we can proceed. Yes, I'm waiting for your confirmation. Okay, only one yes. And what should I take it? How should I take it? I should take it as a no from everyone else. Okay, I hope like this basic meaning of germination is or dormancy is very clear, because they won't be asking you much deeper things in this case of in the case of seed dormancy, but you should have a clear cut understanding between what is seed dormancy and how it is different from quiescent stage. And if in the case they ask you a question where if the seeds are provided with both seeds are provided with both unfavorable environment and favorable environment. If in these two cases, if the seeds are not germinating in case of unfavorable conditions, but they are germinating in case of favorable, what do you call those type of seeds as? These are quiescent. So they can also ask you if the seeds are not germinating in favorable condition. So just tell me one thing: if the seeds are not germinating in the favorable condition, is there any possibility that they will germinate in the unfavorable condition? If the external environment that we are providing in terms of light and temperature and other medium conditions or the media conditions, if they are not even working in the ambient conditions, like in the favorable environment also, if the seeds are not germinating, is there any possibility that they will germinate in the unfavorable environment? I hope like you are getting my question. Yeah, students, you can also unmute and answer me. Okay, no. So this is very clear, right? Because if the seeds in the second case, if they are not germinating in the favorable environment, if they are not germinating, it's pretty obvious that they won't be even germinating in the unfavorable environment. If this is the case, then it is a true dormancy, or these seeds can be classified as dormant seed. So I already told you like what are the reasons for the dormancy, right? I hope you remember from the previous classes what is the main reason for the dormancy. So dormancy, why there is a dormancy in the seeds? Because of incomplete nutrient accumulation, or simply in the broader term, seed is not ready for germination. So in that case, even if you provide the external conditions, even if the external conditions are favorable, but still the internal environment is not fully prepared. So that is why you see this dormancy, and this is why there is a block to germination. So this is how you can understand this seed dormancy, right? So what about the significance, whether this is useful or not? So when we just talk of the usefulness of this dormancy, we have to see the two perspective. One is in perspective of plants, whether they are favorable to plants or useful to plants or not, and second case is for us, that is human beings. So in the first case, tell me like whether they are useful to plants or not. Yes, they are very useful for the plants. I will tell you why, but they will create some problem for human beings, because when you sow the seeds, if all the seeds or of a seed lot they are not germinating, you will see the incomplete plant stand, right? You won't see the complete plant stand. There will be a few seeds which are not germinating, which will ultimately impact the plant stand. That is, plant stand means like number of plants that have been germinated in certain land. So that is how it is affecting it. So if it is affecting the plant stand at the first place, then ultimately it will have the effect on the yield. So even if the dormancy is broken after some time, if this is occurring, for example, this is a seed lot and this is how you are sown the seeds. So because of dormancy, the dormancy is not uniform for all the seeds. It again differs for different environment. The expression of the dormancy will again differs with a different environment that you are providing, because they are ultimately governed by certain genes. So you people know that if it is governed by certain genes, there will be certain environmental variations that you get to observe, right? So that is why if you just have the uniform seed lot, the dormancy won't be uniform. For example, if the first seed will germinate, second will germinate, third won't be germinating, fourth will germinate, fifth this won't be germinating. So this is how the field looks if there is a dormancy in the seed lot. So if this is occurring, so in the later part of the plant development, if they are going to develop or if the dormancy is broken, so is it possible to harvest the seeds at one time? Can you use the machine harvest for the same and harvest all the seeds? Because for harvesting they are at the different stages. In that case, few seeds will be completely filled, and in the few seeds the seed filling is still taking place. So this is one problem. So much before the harvest, there is a major problem during pollination. So that is a problem in synchronization of flowering. So if you take a field when the seeds are sown in the field, so there should be a synchrony in the flowering so that there will be effective pollination at one time. So this has a major effect on a synchronization. So this is how it is harmful to human beings in terms of aspects of seed production, and also in terms of incomplete yield, and in other cases it is also harmful in case of seed processing. So why do you think, like, how this dormancy will have the effect on the seed processing? See, if I say that seed is dormant, or I'll just abbreviate as DS. If I talk about dormant seed, see, if they are dormant, it simply means that seed is incomplete in nutrients. Because if the complete nutrients accumulation has been taken place in the seeds, it won't be dormant. If it is dormant, it simply means that, okay, there has been incomplete seed filling, or the nutrient reserves are incomplete, or there is a genetic block in it. So if this is the case, you know that the nutrients and all have the effect on enzyme activity. So if ultimately it means that in the dormant seed there will be a very less enzyme activity. So how this low enzyme activity will affect the seed processing? Seed processing in terms of fermentation. So the fermentation is an enzymatic process, so that will have a direct effect. So this is how in seed processing the seed dormancy has a harmful effect. This is directly because of low enzyme activity that is observed in the dormant seed. Only in the germinating seed, since the seed reserves are mobilized, the enzymes get activated, and it all starts with the imbibition of water. So once the water gets imbibed, so enzyme activity will be raised, and this is how it works, and ultimately we see the germination process. So if at the first place this water imbibition itself is blocked either by the embryo or by the seed coat, by the structure of seed coat, if it is hard and it is not able to imbibe the water, how it will activate the enzymes? So this is not possible, right? So this is how it is harmful to the human beings, but in case of plants this is very useful. So can you just tell me the examples of how for the plants the seed dormancy is useful? We can just talk of natural calamities. For example, yes, yes, Archie. Yeah, you want to say something? Okay, fine. So you just think of natural calamities that will be occurring frequently or infrequently. So if this is taking place in the environment, so if there is a certain stage where the seeds are matured and they are at the stage that where the dispersal will takes place. So if at that particular condition, if there is some natural calamities and where all the seeds will get destroyed, so if this is naturally occurring, what will be the survival ability of the seeds from generation to generation? We'll just see this in a long run. If this is the case where because of natural calamities the seeds are getting destroyed, so what will be the survival capability of that particular species or that particular seed? So it eventually decreases from time to time, right? So because of this dormancy, because of the variation in the germination in the single plant itself of the different seeds, so because of that they will spread across the time and space. That means some will germinate today, some might germinate after next week, and this is how they will spread with time and space. So this is the major ecological significance of the dormancy. That is, it helps in the spread of germination, and also it relates with the what you call risk of death of unfavorable environment. When the environment is not favorable, so at that time, if they are in the soil, they won't be able to germinate. Eventually, it will die. But if they are dormant in that particular condition of unfavorable environment, so once the favorable environment recovers, so they can germinate. So this is how they can survive, or this is how they have developed the mechanisms to strive or to survive in these hard conditions. So thirdly, they buy the time for dispersal. They will buy some time so that there will be a spread of germination. So by the time for dispersal, when I talk of dispersal of the seeds, you have several mechanisms. So major one are anemochory and zoochory. So I think you have already also heard about chory and phylly. So what is the difference between phylly and chory? Like zoophily and zoochory. So what is the difference between chory and phylly here? Yes. So what will be the difference between chory and phylly? Pollen and seed. Very good. See, the dispersal of seed. Just give me a second. Okay, sorry for the disturbance. So when we talk of dispersal of seed, it is a chory. So don't get confused with chory and phylly. When we talk of dispersal of seeds, it is zoochory. So if we are talking of dispersal of pollen, then it is phylly. So what is zoophily? By animals. If the dispersal is because of the animals, whether it is seed or pollen, if it is by for the seed, then it is called as chory. If it is for the pollen, it is phylly. So this is zoochory. What about anemochory? What is the dispersal agent here in case of anemochory? Yes, fast. I know you people know it. Yes, very good. So it is by air or wind, right? So just try to remember all these things, and I have also mentioned all the different types of dispersal mechanisms in crisp notes that will be providing it by the latest by this weekend. So just make sure to remember them or revise them from time to time, okay, because they might be asking you these things in a direct way. So you should not lose any of the marks from those things. So I hope like this is clear, the chory and phylly. And last fourth one is they prevent the precocious germination. So none of you have reminded me about this precocious germination even after 5 classes. Even after 4 classes, I told you that I will explain what is precocious germination in one of the classes. And there are two different types of precocious germination. One is post harvest sprouting, second one is vivipary. So, and I also asked you to find out the difference between the two. So, but none of you have reminded it. Why? You people have forgot it, and or you people feel that this is not at all important, or this is not under your syllabus, or you just simply neglected it. Can anyone tell me, like, if you have found out, like, what is the difference between pre-harvest sprouting and vivipary? Okay, no problem. We can discuss it. Okay. So this is about prevention of precocious germination. I hope like you people know about what is precocious germination. When the germination is taking place, when the seeds are still on the mother plant, this is called as precocious germination. So it should be avoided, right? Because if they are germinating itself on the mother plant, there won't be much development of the seeds when they are again transplanted into the soil. So that is why this precocious germination should be prevented, and the seeds have inherent mechanisms to prevent this precocious germination. And fifth one is it only favors the germination in the favorable times. That is, partially imbibed seeds will, and there is a other case where the partially imbibed seed. In the first case, if I consider that if you sow the seeds For some time they have imbibed the water. For some time they have imbibed the water. Even after that case, even there is a partial activation of the enzymes, it can again go into a state of dormancy. So this type of dormancy is called as secondary dormancy. So I believe like you will be seeing a lot of terms regarding the dormancy. For example, physiological dormancy, embryo dormancy, seed coat dormancy, physical dormancy, morphological dormancy, endodormancy, paradormancy, ecodormancy. But I feel like you will be not knowing the difference between all these things, right? So though like most of the things are not important in depth, but at least you should know the meaning of all these terms. So this is not available in a single book or at a single place, but though I will be explaining all these things in a simple way. Okay. So now I have explained you what is secondary dormancy. Secondary dormancy is for those type of seeds or those class of seeds where they are not dormant by innate. They are not dormant. See, they have the capacity to imbibe the water. See, if they are imbibing the water, it means that they have a capability to germinate. So if this, even in this case, if they are again going into that period of dormancy, those type of seeds are called as, or those type of dormancy is called as secondary dormancy, and seeds are secondary dormant seed, right? So these are the biological or ecological significance of the dormancy. So lastly, it is about the computation. Yes, of course, if the all the seeds are not germinating at the same time, there will be a lack of nutrient sharing, right? It can save or the plant, like single plant, if it is establishing out of two, that can utilize all the nutrients. But if there are two plants at the same place, so they will compete for the nutrients. So this is how if one is being dormant and one is germinating, this is how it will reduce the competition between the seedlings, and one can develop rapidly. And after some time, once the nutrients are again added to the soil, so other can germinate. So this is how it works. So I have just talked about the benefit of the dormancy. So what about the problems in the dormancy that you see, which I have explained it earlier? One is non-uniform germination, which will have the effect on the synchrony. Second one is problem in harvesting. You can't harvest all the seeds of a lot if there is a dormancy, because they will be at the different stages of their development. Thirdly, from the seed production point of view, there are several difficulties. One is synchronization. So at the time of pollination, when you have to do the ant pollination or the manual pollination, because of non-synchronous nature, so the pollination won't be effective, or you have to do the multiple pollination. So this is again laborious process, right? So this is the one drawback of it. Yeah, I will explain the pHs, not to worry about it. So synchronization is fine. And next is volunteer plants. So how do you define this volunteer plants? So this volunteer plants in simple terms means that, so any type of plant in the field which comes with no effort, without any human effort, if they are developing, so they are called as volunteer plants. It doesn't mean that you sow the seeds and after that you are not giving any effort. Even from the sowing of seeds, you will be not involving anything. That means they will be already present in the field itself. You are not doing anything, but they are developing as a plant. So those type of plants are called as volunteer plants. How do these volunteer plants develop or germinate? It is because of the previous season, the seeds, the harvested seeds might be left over in the field, and they might have undergone the period of dormancy, and by the next season they might germinate. So if you are using the different varieties from the previous class or the previous season, so that time it will again make a physical impurity, or the physical purity of the seeds will be affected. So this is the problem of dormancy in case of volunteer plants. And the laborious to break the dormancy, if in the field conditions, if there is a dormancy in the seeds, so you need to apply the seed treatment. So that is again laborious process. So this is one of the problem of dormancy. And even in the seed testing, if you are doing the germination testing, so germination testing, why we are doing? We are just seeing the ability of the seed to germinate. So we are what you call evaluating the seed quality. So even though it has the ability to germinate, but it won't germinate because of dormancy at the time of germination testing. So if that is the case, for example, the seed lot is fine. I will take a sample of seeds from certain seed lot. Seed lot is actually fine. It is genetically pure, physically pure, and all the seed quality attributes are fine. But I will take a sample and I will do a germination test. For example, germination testing, it is of 400 seeds, 100 into 4 replications. So if in one replication I sow the 100 seeds, just because of dormancy, if only the 50 of the seeds are germinating in all the 4 different replication on an average, so dormancy is 50% or germination is 50%. But actually this is not true, because other seeds have also have the ability to germinate, but in the meantime when the germination testing is taking place, they are not germinating. It doesn't mean that these are not quality seeds, right? So this is affecting the evaluation process in the seed testing, the dormancy. So these are the some major concerns about dormancy. Clear? Just reply with yes if the things are clear of whatever I have discussed till now. Okay. So this is about the dormancy. I will talk about the terms, different terms that will be dealt in this classification only. But I hope like three important things are clear. One is meaning, second one is their benefits and their harmful effects. Fine. So if they ask anything related to three different topics or these three different concepts, I hope like you all will be able to answer them, right? So next we'll talk about classification of dormancy. So when you have to classify the dormancy, there are several categories of how you can do it. For example, in the first case, based on the seed plant or the location of dormancy, you can divide the dormancy into two different types. So one is embryo dormancy. So what do you understand by the embryo dormancy? Dormancy is mainly because of the structure of the embryo. If this is the case, then it is called as embryo dormancy. And secondly, dormancy can be imposed by the coat, seed coat, or the structure of the seed coat. In that case, the dormancy will be called as coat imposed dormancy. But out of these two, if the dormancy is because of the embryo, or embryo means it is because of morpho-developmental immaturity. It is just because of the seed development process the dormancy has occurred. So if this is the case, if they develop some ability to, or if they develop the capacity to grow later or develop later, so in that case this dormancy can be reverted into germination, or dormant seeds can be now called as viable seeds. Why? Because in case of embryo dormancy, it is because of the developmental inadequacies, right? So there is some block in the development. If the after some time, if that block is removed, so they will be able to germinate. That is, they can be converted into dormant seed into viable seed. Viable seed, nothing but they are able to germinate. And even in the viable seeds, again, if there is some inadequacies, they can be again converted into dormant seeds. So which we call it as secondary dormancy. So both the things are possible for embryo dormancy. But in the second case, if the seed dormancy is by the extra embryonal structures or the surrounding structure of the embryo or the seed coat or the testa, if I scarify the seed, I hope like you know the process of scarification. This is just by rubbing the seed coat, because the seed coat will be generally hard in case of legumes. This is the main cause of dormancy, right? So if you just scarify the seeds, it can be permeable to the water. So earlier it will be water impermeable because of the seed coat structure. So if you just scarify the seed coat structures, then it will be permeable to the water. So if they are becoming permeable to water, the enzyme activity will rise and they will be able to germinate. But if I tell you like once this is occurring, is there any possibility that again the seed coat structure can become hard and they will lose the ability to imbibe the water? Is this possible? So once the scarification process is completely done, or any kind of process which is actually based on the seed coat, so if that block is removed, is there any possibility that again this water like they will be impermeable to the water after becoming permeable? I hope like you got this point. Yes, just reply with yes or no, like if you have got. if you have understood like what I've explained. Okay. So see, students, like if you are getting something, please understand, like please reply it immediately, because if you're not doing it, like it's just a waste of time. So if you are replying something, that I at least explain it again, or I can just move forward, right? Okay. So the next thing is classification. I told you about the embryo dormancy and court imposed dormancy, and this is why I have mentioned it as irreversible, because once it becomes permeable to the water, if it is because of the seed coat structures, so then by the action of scarification, the seed coat barriers will be removed. So once the barriers are mechanically or manually removed, you cannot incorporate those barriers again to the seed. So that is why it is broadly called as irreversible case. Not all type of seed coat dormancy, seed imposed dormancy are irreversible, but generally they are irreversible because they are regulated by the seed coat. So again, in this seed coat dormancy, you have the three different categories. One is the dormancy because of the endosperm structure, dormancy because of the pericarp, or dormancy because of testa. And these are the specific example of the crops which have the dormancy, which have the particular dormancy. Just try to remember them. If you can do it. So at least I hope, like for the testa, since it is hard seeds comes in legumes, you will be able to remember it, right? So other two things, it's up to you whether you want to remember it or not. Most probably they won't be asking these direct examples, but since the hard seed comes in testa, they can ask you about this. So at least try to remember the third one. And there can be the case where the crops have both embryo and the coat imposed dormancy simultaneously or successively. So those type of seeds are crops are apple and trillium species. So the dormancies can be called as combinational dormancy, CO, combinational dormancy, because they have both seed coat and also the embryo. And also it might be in the simultaneous case or in the same time, or it can be successively. That is one after the other. So this is a one type of classification based on the location of dormancy. So secondly, we can also differentiate based on the time of induction. So primary dormancy or the innate dormancy, or the secondary dormancy or the induced dormancy. I hope like you are able to see the different terms that I am using here. See, there are again three different terms here. One is induced, innate, and the primary dormancy. So primary and the innate dormancy are one and the same, and secondary dormancy I have already explained, and this is same to induced dormancy. So in case of primary dormancy, the primary dormancy induction is at the maturity when it is on the mother plant. And again, the primary dormancy, what can be the reasons for the primary dormancy? It is either by the embryo or the seed coat, which we have discussed it earlier. So a lot of things overlap in the categories, but it is based on the different types. Like for example, one is based on time, one is based on location. So this is how they are differentiated, but the meaning remains the same. So one is primary, second one is secondary. Secondary is also called as induced because the non-dormant seeds will be forced to go back to the dormancy. So again, in the case of secondary dormancy, we have three major types. So have you heard of scotodormancy, thermodormancy, and complex patterns? Complex patterns, I hope like already you got this point. Complex means it will have several depths of dormancy or different depth of dormancy. This is why it is a complex pattern. But what about scoto and thermal dormancy? So what do you understand by scoto and thermo? I think you can decode this. Scotodormancy is something that is related to light. Thermodormancy is something which is related to temperature. Yes, Archie has answered it right. It is light and temperature. Scoto, it is related to light, and thermodormancy, it is related to temperature. So scotodormancy is something where the imbibed seeds require the light for germination. In the dark conditions, if they do not germinate, then they are called as scotodormant seeds because they require the light for germination. But in case of thermodormancy, it is only like it exhibits the dormancy when they are exposed to the high temperature. So don't get confused again here. See, if they are exposed to high temperature, they are not germinating. They are actually becoming dormant. In case of when they are exposed to high temperature, there will be a germination block within the seeds. So that will eventually lead into the dormant state of the seeds. So this is for thermodormancy, but it is exactly opposite here. When you provide the dark or the light conditions, so then only it will germinate or the seed will germinate. Otherwise, in the dark conditions, they are dormant. So this is the difference or the meaning of scotodormancy and the thermodormancy. And there is also one separate category which is a complex pattern. Why? Because we know that embryo has different parts or seed has a different parts. So the different parts of a seed or the embryo will show the different depths of dormancy or the different patterns of dormancy. So that is why it is a complex pattern, and this is generally observed in Lilium species. Yes, the terms are clear. I just talked about what are the different terms I talked about: primary dormancy, secondary dormancy, innate dormancy, imposed dormancy. Fifth one, scotodormancy. Next was thermodormancy, and also about quiescent state. Is this clear? And also about eighth one, embryo dormancy and coat imposed dormancy. So this is all about the terms of for the different categories. So now, well, if you ask me, okay, there are several categories. So what is actual classification of dormancy, which will cover all the aspects or all the mechanism of dormancy? If you ask me like that, or if you get this question, so since it is very complex, you will definitely get this question. So how do we actually classify this dormancy, or don't we have a system where we have all the different cases of the dormancy at one place? If you ask me that, there is a classification which has evolved from time to time, where classification of dormancy has 4 different systems, out of which the latest one is accepted. So this is how it went. So first in the Harper, he gave one system in 1957, and the Langs gave one system in 1987, and next it was Nicolena. He gave the system in 1969 and 1977. He revised it, and lastly it's a Baskin and Baskin, which was given in the 2004. So this is all from the corrections from the previous classification. So this is the one which is currently working, which covers all the different mechanisms of the dormancy. Okay, so it is a three tier system. This is the spelling is 3-tier system of Baskin and Baskin classification, where it will have the classes, levels, and I hope like it is section. I'll just confirm it once. So these are the 3 different tiers under the Baskin and Baskin classification, where these are the 5 classes. Class 1 is physiological dormancy, second one is morphological, third one is morphophysiological, fourth one is physical dormancy, fifth one is combinational dormancy. Again, within this class, you will get to see the level. Within the level, there will be a section. So this is how it is classified by Baskin and Baskin, and this is the most recent one, and this is currently working. So you need not to worry about the scientist, and we need not to worry about all the different types of systems that I have written it previously. But for just a general idea of how they are classified, I've all mentioned all these names. Okay, so this is about the classification which is actually required for you. So next we can talk of the mechanism, how actually the embryo dormancy occurs, or what are the factors that is contributing for the dormancy in case of embryo dormancy. So it's mainly because of the germination blocks within the embryo, the blocks that are present in the embryo which will inhibit the germination. So the blocks can be in terms of undifferentiated embryo, and this is seen in the Orchidaceae, that is which contains no endosperms, and since no endosperm, no food resource, and the cotyledons has not developed the ability, if they have not developed the ability to store the food, in that case you will see the undifferentiated embryo, and this is the common case of Orchidaceae. And second one, underdeveloped embryo. So even if the differentiation of the embryo has taken place, but it is not fully developed. So immature embryo can be the result of the embryo dormancy. And the third one, there might be the chemical inhibitors, in which the main chemical inhibitor is ABA, which is abscisic acid. So this will have the major role to play in case of seed dormancy, or major role to play in case of switching between germination to dormant state and dormant to germination state or viable state. This is, I told you, this is because of two important hormones. One is GA and ABA. If the GA levels are high, the equally the ABA levels will be low. When the ABA levels are at high, the GA levels will be low. I also showed this in a one practical example of with a one figure in the previous class. I hope like you people remember it, right? So this is about the chemical inhibitors. So there can be also the physiological constraints, where there is a specific balance of GA and ABA that should be there in the seeds in order to germinate, or in order to the dormancy takes place. So this is controlled by the physiological process of the seed, either in terms of enzyme activity or production of the hormones or production of the different proteins or mobilization of seed resource. Any type of physiological event, it will have the effect on the embryo. So this is how the embryo dormancy is affected by all these factors. So broadly, if I tell you about what are the factors mainly responsible or mainly affect this dormancy, one is genetic factor, which I told you earlier, which I used as an internal environment, right? The second one is environment. So genetic and the environment are the two important factors which will affect or which has an impact on the dormancy. So what are the different environmental factors that are actually responsible or which will have the effect on the dormancy? I think I told you about the temperature already, the light, and third one is the media or the soil conditions. So all these things will affect the dormancy in one or the other way. So how it will affect, I just noted all the things here. You can just go through the text once. If you don't understand anything, you can just come back to me, because these are all self-explanatory. You can understand by yourself. These are just the facts that I need not to explain it anything. So okay, we have just talked about the classification and the different factors affecting it. So what are the factors that will have the impact on the release of dormancy? If the dormancy is to be released, so what are the factors that are actually impacting this release? So mainly it is because of the hormones, and secondly it is the after ripening process. Can anyone tell me like what do you understand by this after ripening? And I'm very sorry to bore you with the same type of words again and again. Can you tell me for everything? But I just wanted to know the answers from your side so that I can keep you engaged in the class. See, the after ripening is a process of a seed maturation that will takes place when the seeds are shed, or when they are separated from the mother plants. All the different developmental and the maturation process that will occur in the seed, this is called as after ripening process. So the environment on which this after ripening process is occurring, or how it is occurring, will affect the release of dormancy. And next again, it is the temperature, light, and the different chemicals. And now we will be talking about the most important topic for your exam point of view, that is dormancy breaking methods. So you can definitely expect the questions from dormancy breaking methods. Even if they have not asked in any of the questions in 2022, there is a high possibility that they will ask you something regarding the dormancy breaking methods, since they are very important. So there is a broad classification of, or broad and simple classification of dormancy breaking methods. One is physical method, second one is physiological methods, and third one is chemical methods. So when I talk of physical methods, it is by just manual scarification. Again, scarification can be of manual type, acid scarification, and bioscarification. Bioscarification is nothing but it is with the help of animals, insects, or microorganisms. and these are the examples. When I talk of acid scarification, it is mainly by the sulfuric acid that is commonly used to break the dormancy, which is called as acid scarification. So that is 100 ml of the H2SO4 is per kg is generally used for 2 to 3 minutes, and this is how the dormancy is broken. Or manual scarification is just by rubbing the seeds or using the sandpaper or other things. So these are the some physical methods which will help in breaking this dormancy. And secondly, in the physical methods, we have the electrical seed treatment. Even if we give this electrical seed treatment, so there is a chance of activating the enzymes, and this is how it can break the dormancy. So this is the second case. Thirdly, just by soaking of water. So, and it's all depend on the enzyme activity. The enzyme activity is favored or triggered by the soaking of water. If you just soak the seeds by overnight, so even the seed coat is hard, eventually over time it will be spongy, and it will slowly allow the seeds to imbibe the water. So this is how you can break the dormancy if you just immerse the seeds in the water or soak the seeds in the water for at least 28 to 48 hours. How? Just because of activating the enzymes. So germination process will take place. So next is physiological methods. So one of your friends has asked about the stratification in the WhatsApp group. So I've clearly explained the difference between the stratification and what actual how the actual stratification is takes place, and also difference between cold and warm stratification. So you can just again read the same text to understand this. And the warm stratification, it's commonly used in case of cashew. So lastly, we have some chemical methods. The chemical methods includes the growth regulators, that is GA, ethylene, are commonly used to induce the germination or to break the dormancy. And fusicokin, this is a toxin that is produced by the fungus. So this is also used to break this dormancy. And lastly, it is soaking in the chemicals. The different chemicals are like most importantly thiourea, hydrogen peroxide, and the different alcohol solutions, and also KNO3, that is potassium. This potassium nitrate, and this is recommended by the ISTA. So this is the method to break the dormancy which is recommended by the ISTA, which is of 0.2%. So out of curiosity, I'll just ask you, can anyone tell me like what is ISTA or full form of ISTA? ISTA is the major organization for seed production or seed industry global, for global seed industry. Yes, okay, very good, you at least know about these things. It is international seed testing, not agency, it is association. So it is international seed testing association, where it was established in 1924 with the motto that its main motto or the principle is bringing the uniformity in seed testing procedures globally. So this is the main motto of the ISTA, and it has its own manual called as ISTA manual, which is the basic reference material for the seed testing process and the procedures. So that is actually followed globally. For example, so if you want to check the seed quality or check the quality of the seed, so how you will do it? You will either test the physical purity, or you will either check the germination, or you will just check the genetic purity of the seeds. So this is how you determine the quality of the seed. So the methods to check this genetic purity, physical purity, and also moisture content, germination differs from region to region and for country to country, right? So when you just do it in a different methods, it is near to impossible to compare those different results of those different methods, because at the first place that methods are itself different. So how can you compare the result between them? It is just comparing the results of JRF with the neat results and the JRF results. So this is how it looks like. So to bring up the best possible methods which should be actually used for the specific crop, it is a crop specific. It's not like generalized. it is crop specific. For every crop there is certain prescribed method by the ISTA how we have to do it. So this is how it works. So since the ISTA has prescribed the methods, standard methods for different crops, so globally if you just want to compare this quality of the seeds from two different regions, you can go through the one single method and you can just um, compare the quality. Someone wants to ask anything? Like one has raised the hands. Okay. So next is about cold stratification. The main difference is in terms of the temperature. The cold itself, it is at the temperature of 1 to 5 degrees Celsius, and when you talk of warm stratification, it is of 20 to 30. And other things, it will generally break this cold stratification or the cold condition, break this physiological or morphological dormancy, but the high temperature or 20 to 23 will break the physical, mechanical, and the morphological. So this is the difference between the cold and the warm stratification. It is mainly in terms of the temperature difference. So the one is 1 to 5 and secondly it is 20 to 30. So this is for today's class, that is seed dormancy. So I just wanted to make it very simple and concise, not dragging it too much. So that is why I will be taking for only 1 hour. And also, even if it takes more than 1 week, I will definitely take the classes and I will finish the syllabus. Don't worry about it. And also don't stress about, don't take much stress to watch these videos at a one stretch. So that is why I'm making it very small. So we can again take up the new topic tomorrow and we can discuss it in a detailed form. Okay. So any doubts from today's class? Yeah, the notes will be uploaded by today, but unfortunately I don't have an option to upload the notes for all of you, only to the registered students. Since you have a subscription to the app, I can upload the notes there directly. So like few of you since you are attending these trial classes, I think most of you have already registered to the course again, just by attending these free classes. I hope like you have felt it good. So the students who have not registered our course yet, you have at least 2 months of time from now. So we have very concise and precise videos for you for make your preparation better. So you can definitely consider to take this course now. Don't think that it's already very late to take the courses now. Even if you take now, you will be able to clear all your doubts and you will be very confident about in the subject. You can go and write the exam with confidence. Okay. No for what, Nitish? I didn't get you. No for what? It's no. Okay, fine. Okay, no doubts. Fine. I hope like these seed science and technology classes are making it simple to understand the seed science. Why? Because I have struggled a lot during my JRF period where we don't have one prescribed book. We have several different books, but we cannot study all these books for just to attend the 5 to 10 questions in exam, right? Since the genetics and plant breeding and pathology, most of the questions will be asked from those books or those topics. We can spend a lot of time there, the time and efforts matters there. But since the C technology, the questions will be very less, you cannot stick to more than 3 books and read them all and understand them in first place, and you cannot digest them. So the same problem I have faced it, but hopefully I'm trying my best that you people don't face the same problem. And this is how I am also giving you the notes that I am writing for you. So make sure to use it and just revise the things and at least know the worth of what you are getting right now. Okay. So if you don't have any doubts, we can meet in the next class and we will start our discussion. We will discuss few things about what you call seed mobilization, that is synthesis and mobilization of the nutrients in the seeds. And next we'll talk about the germination, that is apigeal and apogeel, and also about the pre-harvest protein and vivipary. Okay, so this will be dealt in the next class. Once this is done, and I will be shifting my focus on to my favorite subject or favorite field of the seed science and technology, that is seed legislation and certification. I can bet you like you won't be knowing most of the things, or you will be having a lot of doubts in those cases. I will really try my best to bring out all those give all those clarification for all your doubts. Okay, just make sure to attend it and take the full benefit out of it. Okay, we'll meet in the next class. Thanks for attending and good night.